The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, Merry Christmas and welcome to episode 71 of the eLife podcast. I'm Chris Smith from The Naked Scientist. This month, the animals that turn their dinner into solar panels, the first images of anti-nausea drug molecules engaging with their receptors, and what thousands of you told eLife about the people who support their colleagues at work. But first, can exercise cut cancer risk? Various lines of evidence suggest that it can, but how? Randall Johnson has found that the same stuff that makes your muscles burn when you run also stimulates classes of immune cells that can suppress the growth of cancers.
A number of studies over the years have found that there are links between increased exercise and physical fitness and decreased susceptibility to cancer. And there are animal model links that have shown over the years that you can induce animals to exercise. Mice in particular love to exercise. They love to run on wheels and do it voluntarily. And that they will get fewer tumors and the tumors will grow more slowly.
Of course, one possible criticism is that individuals who are more prone to cancer are just lazier.
Yeah, and their lifestyles may be more prone to induce malignancy as well. And it's one of the things that you do these experiments to try and test.
And did you get a sort of handle on that?
What we did is essentially first start out to just in our own systems try and determine whether we saw a decrease in tumor growth when animals were allowed to exercise. And these were, in all cases, animals who just had a free wheel put inside their cages, and they were just allowed to run as much as they want. And mice will run up to 6-7 kilometers a night. And so we found that animals who got a wheel in their cage that was locked, so they couldn't run, had greater tumor growth than animals who were actually allowed to run every night.
Is this a dose-dependent relationship? Because that's one of the other key criteria with cancer causation, isn't it? Is it dose-dependent? So if you allow a mouse to run 6 kilometres, does it get more therapeutic suppression of its cancer than if you restrict it and it's only allowed to do 3 kilometres?
Yes. In our study, there were a group of mice that ran much longer than another group of mice, and we segregated them by rough categories, ones that ran around approximately 3 kilometers a night and ones that ran around 6 kilometers a night. And we did find a dose relationship in that regard. However, we're now doing experiments where we're much more closely controlling the amount that the mice run to try and approach that very question.
And putting some numbers on it, how much of a difference did it make to, say, the volume of tumour in a mouse that exercised versus one that wasn't able to exercise?
Yeah, we were seeing reductions in tumor growth of quite substantial ones, up to 50%. But of course, their cancers weren't being cured. What we seem to see is a link to the role of a specific type of immune cell, the cytotoxic or killer T cell, which in many regards is considered to be a very important controlling element in the immune system to combat cancer. And those cells seem to be much more prevalent in the tumors of animals that exercise.
Your hypothesis being then that exercise in some way mobilises a particular population of white blood cells, these cytotoxic T cells that will take down tumours, and that because there's more of them, the tumour bulk is lower and the animals live longer compared with ones that don't exercise.
I need to be careful. There's not more of them in the animal, but there are more attacking the tumor, so we seem to be potentiating their effect on the tumor. We saw that sort of correlation, but we wanted to test it, so we did an experiment. You just simply kill off all the animals' cytotoxic T cells. Then the exercise-mediated effect goes away.
Which points the finger strongly at those cells as being a key part of the process. What about doing the opposite, where if you collect those T cells and put them into an animal that's not allowed to exercise, so it's a lazy mouse, does it inherit the protection as though it were exercising by doing that?
Yes, you hit the nail on the head. That's exactly what we did next. We basically took T cells from trained animals and put them into non-trained animals. To coin a phrase, could we train the T cells, would they then be better at reducing tumor growth? And in fact, they were.
It's extraordinary. Does this argue then that there is something specific coming out of muscles when they move and when we exercise that causes these cells to increase their activity? Do we understand, or do you understand yet, what is actually driving the link between physical activity and these T cells that seem to have this anti-cancer or cancer-suppressing activity?
We think we have a good handle on it. One of the really exciting things we followed up on was that muscle produces lactic acid, which we know is important, and you feel that in your muscles when you exercise, and that lactic acid in the blood could actually have striking effects on T cells, improve their performance in combating tumors.
And have you actually tested that? Can you stimulate T cells with lactic acid and demonstrate they go into this more enhanced, weaponized anti-cancer mode?
We could. We injected the animals on a daily basis with a quite substantial dose of lactic acid, but enough that we actually were essentially mimicking what would happen from a big bout of exercise. And we saw that when we did that, the high doses actually did reduce tumor growth.
And so do you think that there might be a solution for people who are couch potatoes? I mean, I'm the worst of them. And whereby I can have my cake and eat it, quite literally, and keep my cancer low by either dosing myself with something that will fool my immune system into thinking I've been a lot fitter and less fat than I am, or stealing someone else's T cells and infusing those.
There's that. There's always this sort of Count Dracula mechanisms of eternal youth that are out there. And there are cancer therapies that involve reinfusing T cells. And what we hope is that this may help us find ways to treat those T cells to make them better anti-cancer agents when they're put back into patients. I would say, this is pure speculation, of course, there's a lot of lactate in yogurt. I'm sure you're aware there are these populations in Greece and in the Urals that the centenarians are a very large proportion of the populations, and at least in the yogurt ads, they always say it's because of their yogurt consumption. So it's probably not bad for you to eat more yogurt.
Well, I shall have an extra big portion of Greek yogurt today to help to offset the time I've spent sitting down recording this podcast. That was Randall Johnson. He's at the Karolinska Institute and also at the University of Cambridge. Now, we all know we ought to eat our greens, but for photosynthetic sea slugs, this takes on a whole new meaning. These small marine creatures feed on algae and steal the photosynthetic chloroplasts in the algae as they digest them, incorporating the chloroplasts into specialised pockets in their digestive systems where they can be kept still in a photosynthesizing state for up to a year. Vesa Havarin from the University of Turku has recently found that the sea slugs induce protective changes in the chloroplasts that they steal, and that's what allows them to keep going for so long. Eva Higginbotham heard how it works.
The term sea slug, it refers to a large group of animals. They are gastropods, just like your regular garden snails and slugs, and they can look quite alien, to be frank. But when you're talking about photosynthetic sea slugs, you are referring to a very specific group of slugs, and they are called these sap-sucking sea slugs, or sagoglossans.
You've actually sent me a couple pictures of some sea slugs, so I'm just looking at them here, and they're quite cute.
Well, if you're into that sort of thing, but yeah.
And so they're sort of got a white or sort of opaque-ish white-looking body with some parts of them that are green. Where does the green colour come from?
Yeah, the green color comes from the chloroplast that they steal from the algae they feed on.
So what makes the sea slugs photosynthetic?
Well, they steal the chloroplasts, which are the organelles inside plant cells or algae cells that utilize light energy to convert it to sugars by fixing CO2 from the atmosphere or the surroundings. So basically these slugs, they are taking that organelle from the algae that they eat, and then they those foreign organelles, chloroplasts, inside their own animal cells, and then they basically carry out photosynthesis just like the algae would.
Why do they do that?
Well, usually when people are answering this question, they are stating the obvious, like that the slugs would get a energetic benefit, but it's still debated
So what were you trying to find out in this study?
We were trying to figure out if the slugs induce some protective changes to the chloroplasts they steal from the algae. The thing is that when the chloroplasts are inside the slug cells, they are actually quite isolated. They are, for example, cut off from most of these repair mechanisms that are available to them in their normal environment, meaning the algae cell. So, because photosynthetic machinery of the chloroplast, it's actually constantly being damaged by light, even though light is required to drive photosynthesis, basically. So we were trying to figure out if they somehow reduce the damage to the chloroplast in the first place, so that they wouldn't have to fix so much damage.
And what did you find?
We compared photosynthesis in the algae versus the slugs, and we found out that the slugs do actually induce photoprotective changes to the chloroplast. The first thing that we noticed was that the slugs actually maintain the photosynthetic machinery in so-called emptier state, electron-wise, than the algae. And this allows for more fluent conversion of light energy into sugars and reduces the risk of this excess energy going into unwanted directions, for example, to oxygen, which can create reactive oxygen species and damage the chloroplasts and the slug for that matter. The second thing that we found was that the slugs actually have a more efficient way of preventing excess light energy actually reaching the photosynthetic machinery than the algae. And the third point was that if all these other measures fail, the slugs can still utilize the same safe energy sinks that the algae actually use. So when they are exposed to a very strong light, for example, they can still deal with that excess energy safely without producing reactive oxygen species.
Are photosynthetic sea slugs the only sort of animals that eat plants and steal their chloroplast to use in photosynthesis in this way? Or is this a mechanism that other species have too?
For a long time, it was thought that these sap-sucking sea slugs are the only animals capable of this. But there was a recent paper where they showed that actually there are these marine flatworms that are able to do the same trick. And it would be interesting to get my hands on those as well to see if there are some common characteristics within these two different animal groups. And if there's some general rules for this, stealing chloroplasts and using them for your own gains.
It's an extraordinary story, that, isn't it? Vesa Haverin there. You're listening to the eLife podcast with me, Chris Smith. Still to come, how the brain labels a taste as bad news. But first, a class of drugs called cetrons, one of the best of which is ondansetron, are powerful anti-nausea agents often used in patients with disabling sickness during, for instance, cancer chemotherapy or radiotherapy. Treatments like this lead to higher than normal levels of the signalling molecule serotonin in the blood, which can activate a region in the brainstem that triggers vomiting. The cetron drug family block one class of serotonin receptors to stop this happening, but their interaction with this receptor has never been visualised directly. It's only ever been inferred, and this means we don't know if we could possibly make modified cetron molecules that would work even better. So Sudha Chakrapani from Case Western Reserve University set out to capture detailed images under the electron microscope of this happening.
Based on the chemical properties of cetrons, it was known that it is likely to bind in the same pocket as that of serotonin. And there were indirect studies that showed that it is likely to bind because you make certain perturbation to that pocket. It also not only affects serotonin binding, it also affects citron binding. So these were all indirect indications that citrons bind in the pocket, but it was not shown experimentally. So why this is important is because even subtle changes to the citron molecule can have a huge impact on the efficacy of the drug or the effectiveness of the drug. So these are the informations that were not available before the study.
What did you do then to turn what was indirect evidence into direct visualization of when these drug molecules go in, where they bind to, and how they exert their effect?
We used an approach called cryo-electron microscopy, isolated the receptor. We pick out individual particles and we align them and we create a three-dimensional reconstruction of this molecule. And we did this in the absence of citrons, in the presence of serotonin, and in the presence of different types of citrons. So we were able to identify the interaction fingerprint, as you may say, between the drug and the receptor.
So you're literally able to see the molecules themselves and what the molecule looks like when serotonin's in there, when the drug's in there, when nothing's in there, and you can see how the shape of the molecule can be distorted or changed when those interactions occur.
Exactly.
The crucial question, of course, must be that when you do this and you can really see for real what was happening when the drug was present, does the direct visualization tie up with the indirect evidence of how you thought these drugs were working? And what did you learn?
Yes, the findings agreed very well with what we knew from findings in the past. And there were a few surprises. We were able to identify certain citron orientations ended up being slightly different from what were predicted in the past. And we were also able to rationalize what could have been the causes for this discrepancy between past findings and what we are finding right now.
And is that the crucial step that will then enable us to optimize those drugs going forward?
Yes, absolutely. Having a much better idea about what are the bases for the drug receptor interactions will definitely pave the way for using this information to design better drugs.
Obviously, when we tweak any drug or we try to make a drug, we have to be very cautious about off-target effects. Is there a danger, based on your knowledge of these sorts of receptors and these sorts of drugs, that in trying to optimize the drug better for that particular receptor, we might end up making it bind better elsewhere as well and therefore causing more side effects?
Absolutely. This is a danger when we try to modify the drug, and actually, we are only looking at one target at a given time. So we have to tread very carefully. One way is there are many different types of serotonin receptors that are present in the body. They differ in their location, let's say in the brain versus in the gut. So it is important to understand the functioning of each of these type of receptor, and a detailed knowledge of those will allow us to eventually design drugs that are specific for one type versus the other.
Sudha Chakrapani. Memories of previous bad food experiences that make us ill tend to form fast, and they also tend to endure. Going back decades, scientists had shown that two regions of the brain, one concerned with taste and the other concerned with fear and emotion, are tightly connected, and forming these avoidance memories depends on both, although the exact wiring arrangement wasn't known. Now, Ariana Maffei has found the surprising result that when something makes us sick, the strength of the connections between the two key brain regions loosens. The findings are far-reaching, from helping patients undergoing chemotherapy in hospital to conserving endangered species.
The circuit we looked at is the connection between the gustatory cortex, which is the area of the brain that processes taste. Taste is intrinsically good or bad, in addition to having its own identity, whether it's sugar or salt. And the amygdala, which is a center of the brain that is known to process positive and negative emotions. So we know from previous work that these two areas are involved in assigning a positive or negative value to a taste stimulus, and we wanted to know how that assignment happens.
So in other words, if I had something like an ice cream and I ate the ice cream and I concluded I liked it, versus if I have something that I definitely didn't like, the assignment of niceness or nastiness is bound up in this part of the brain, the gustatory cortex and its connections to the adjacent amygdala.
Yes, that's exactly how it would work. So the amygdala would assign a pleasant or unpleasant flag to the taste, to the ice cream.
But what happens if I overdo the ice cream and I end up being sick? And then afterwards, I tend to avoid ice cream because I'm thinking I don't like it anymore because it made me sick. Is that circuit bit the one that's being changed here?
So this is exactly what we sought out to find. And our hypothesis was that, yes, there is a modification in the connection between the amygdala and the gustatory cortex when somebody eats something that makes them sick. And this very strong memory, known as condition taste aversion learning, is formed. So we really wanted to know how neurons form this memory and how this memory gets stuck in this circuit.
So where did you begin?
So we began by inducing this form of learning in laboratory animals. We used rats. So what we did is we offered our rats sugared water that they really, really enjoyed to drink. And then we associated this sugared water with an injection of a substance, lithium chloride, that made them have a bellyache. And then the next day, we offered them a choice between the sugared water and just plain water, and we wanted to see if their preference for sugar had changed. And in fact, they drank a lot more water than sugared water on testing day when the association had been formed.
And your conclusion from that part of the experiment would be, right, we've got this connection in the gustatory, the taste area of the brain, which originally was assigned an I like this message. And after you make the animals feel unwell for a short while, that I like this has been turned into an I don't like this signal. And you're presuming it's some kind of connection between the amygdala and the taste center, the gustatory cortex, has changed.
Yes, so we know that memory has formed, and from previous work where people were making small lesions, we started out with the hypothesis that the amygdala and the gustatory cortex are necessary, and we wanted to see what happens in the connection between them.
And if you go in and sample those two areas of the brain, can you see, either electrically or chemically, any kind of difference in the behaviour of those sites after you've changed the way the animals regard the sugar water, so that once the animals start to avoid it because they don't like it anymore, does the wiring change?
Yes, we went to look into whether the connection had changed, and we did find a change. What we did find was a surprising change, because the hypothesis typically is that when a memory is formed, the connection between two areas or two neurons strengthens. What we did find instead is that the connection between the amygdala and the gustatory cortex had become weaker. So it was exactly the opposite of what we had predicted.
Now, if that's the case, is it possible to simulate this aversive response by inhibiting or turning off the connections from the amygdala without any kind of making the animals feel ill? So if you just go in and artificially switch off those connections, can you get the same aversive response?
We try that by using a tool that is called optogenetics. It allows us to activate very selectively only a specific connection in the brain. And we implanted an optic fiber in the gustatory cortex, and we tried to make the association between the sugared water and activity that would decrease the strength of the connection between these two areas directly in the gustatory cortex. And indeed, what we found is that we could completely substitute the bellyache with this stimulation directly in the brain, and the next day the animals would have learned not to drink the sugared water.
And why does this matter, the fact that you found this? Because going back to the 1950s or so, people had made discrete lesions in brains in these areas, and they demonstrated that there is a loosening of this association. If you do that, you can prevent this effect happening. So we had an inkling that this was going on. You've confirmed it and proved that that's what's going on independently and via a different way of doing it. But beyond that, are there other implications?
Yes, it is very important to know how these form of memories are formed from the intellectual perspective, just to know what is the signature of learning in the brain. But from the more practical perspective, for example, we have patients that are undergoing chemotherapy that, as chemotherapy gives them a bellyache in many cases, it can actually form an association between the food that they have eaten before and giving them a really hard time being able to self-sustain and eat the food that they previously liked because of the therapy. So understanding how this form of memory works also tell us strategies to avoid these negative associations in patients, for example. Another reason for why this is interesting is that this form of learning is conserved across many, many species. And in conservation biology, ideas that are related to condition taste aversion have been applied to try to protect endangered species from predators. For example, by including the emetic agent like the lithium chloride in the eggs of the species that they want to preserve so that the predator will be conditioned not to go eat them.
Stony Brook University's Ariana Maffei. An emerging trend in academic settings is mental health. Increasing numbers of researchers up and down the career ladder are disclosing mental health problems. Now, this could be because the situation is worsening or that the working environment is becoming more receptive and supportive in this area. Or it's both. But one thing that's often not being discussed is the people to whom those who need support are turning. Who are those supporters? And who helps them? Is this an organised system or an ad hoc one where people are taken under a nearby sympathetic wing? And are these supporters properly equipped to guide their colleagues? Are their efforts recognised? Surprisingly, the answers to all of these questions is we don't know. So eLife's Elsa Loisel decided to set up a survey to try to find out. In her words, she humbly expected a handful of responses. She got over 2,500.
We have increasing evidence that there is an issue with poor mental health in academic settings and that supporting relationships are quite important. What we wanted to know and what is less discussed is what are the people who are supporting others feeling and what do they need?
When you say we've got evidence that there is a problem, what is that evidence and how extensive is it? And is it getting worse or has it just always been there and people are getting better at talking about it?
That's always the question. So there is a lot of sort of anecdotal evidence. There are more studies coming out showing the rates of mental health issues within graduate students is higher than it is in the general population or comparable groups in the general population. The amount of stress in staff is comparable to healthcare workers, for example. In terms of getting worse, there's a report that's been published showing that staff is trying to access counselling services more and more. So in general, it sounds like there is a problem in academia with mental health.
So what did you actually do here in order to try to find out the extent of the problem and really what the problems are?
So we collaborated with researchers to design a survey that would be a snapshot of who these supporters are, what they do, who do they support, how they feel, what do they need. I thought we would get a few hundred answers and we could write a nice little feature article about it. And in the end, we got nearly 2,500 people who started the survey. And the final data set, that's about 1,900 people in total.
Of course, any kind of study has limitations. And when it's a self-selecting survey, there are also going to be biases because of who chooses to fill it in. So notwithstanding that, what do you think the strengths and weaknesses of what you've got here are?
We have not captured the experiences of all supporters. We cannot say anything about this happens at, this rate in the community, or we cannot say anything about these people are more likely to be supporters and these people are less likely to be supporters, because it is a self-selecting survey. So the people that took it were people that understood the message, were interested in mental health, had experiences of being supporters that they felt strongly about reporting, strongly enough that they spent 15 minutes filling a survey about it. So this sample is not representative.
And what did they tell you when you went through the responses? What were the key findings, the key trends that kept emerging again and again and again?
A lot of the support was taking place between peers or between a senior researcher that was not officially in charge of the junior researcher that they were helping. So it's a lot of potentially invisible work that is being done.
And that's obviously not being recognised. It's not being rewarded either, I presume.
No, and that's also one of the messages that really emerged. Many supporters need support themselves, and they don't receive it from institutions. I think there is a quote that summarizes brilliantly. Someone that says, institutions love us supporting students, but they don't value it, they don't reward it, they don't take it in consideration when they look at workload models or promotion criteria.
What other important trends or findings emerged?
women emerged as a population that faced quite a lot of additional challenges. So they were more likely than men to support more than one person at a time, to feel that more people were coming to them because they were supporters, to feel like being a supporter affected their work and left them stressed, took a lot of their time. Early career group leaders also were a population that felt they needed support in their role, and they felt that their work was affected by being a supporter. There was also a huge overlap between people who were struggling with their own mental health while they were helping someone else. The majority of supporters also felt very positive about having helped someone, that they found it meaningful, that they felt that they made a difference in someone's life. And so I would hate to paint this as a burden or as a chore. This is something that people felt very strongly about doing, and they felt natural doing, and they felt distressed not being able to do properly.
And to finish, what do we do about this then?
Well, I think at the level of just the individual, I think for people who are supported, there was a very strong response from supporters saying that they believe that these people belonged in science, that they believe that they were good scientists regardless of their mental health. And so sort of reassurance that they are worthy of being there and of being helped. And I think for supporters, there was a sense of isolation coming from some of the open answers, and feeling that there are others that are going through those potentially difficulties of not knowing what to do, and sort of maybe open discussions within the lab saying, oh, my student is having not a great time, what do you think I should be doing? At the higher level, for institutions, understand what is going on within their department, then they can tailor solutions that would really help the people that are doing the work already. We need to understand those relationships better, and that's why we've made our data set and our code available, because we've barely scratched the surface here, and there are people out there who could use this data set and this code and build on it and explore it even further.
An excellent starting point. Elsa Loisel there. Well, that's it for this month, and also for this year. You've been listening to the eLife podcast. It's produced by The Naked Scientists, and previous editions, references and the full text transcripts for these programmes, as well as details on how you can subscribe to this podcast, are at nakedscientist.com/eLife. Incidentally, The Naked Scientists also publish a weekly science programme that covers the latest leading science stories. You can find that at nakedscientist.com/podcast. I will be back in 2021 for another look inside eLife. Until then, from me, Chris Smith, have a wonderful Christmas and New Year. Thank you for listening, and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.